Our next speaker is Dr. Muhammad Zahir Iqbal. Dr. Iqbal got his PhD in 2014 in physics from Sejong University, South Korea. And then he went for a postdoc to Georgia State University in the USA. He is working currently as an associate professor of physics at GIK Institute in Pakistan. He has been very productive. He has already published more than 50 papers, and he will tell us today about his work on nano devices for biosensing and bone engineering. So, Dr. Iqbal, if you could please go ahead and share your slides with us and take it over.
Assalamu alaikum and good morning, everyone. Can you hear me?
Yes, we can hear you.
Okay. Thank you very much for the nice introduction. Okay. I will discuss today about because this conference this workshop is diverted towards biosensing. So I will focus my talk to for biosensing, and my topic of today's talk is two-dimensional materials based biosensors and BioFETs. I am working here at GIK, and I have my own lab with the name of Nanotechnology Research Lab. Also, we have another group with the name of STEM Nexus, in which we are working together with some other collaborators from the mathematics department, and some from the engineering department, and some from the kind of industry, and we are actually doing kind of a collaborative work. And this group is we are running with the name of STEM Nexus. So basically, I am working in a various field, as the professor introduced me, that I am working with the field of biosensing, and but it is one of the one field in which I am working, but I am also working in some other fields like two-dimensional materials based devices for various applications such as spintronic devices, quantum devices, electrochemical sensors, optical sensors. Recently, we have started work on the supercapacitors and batteries and kind of a supercapacitor. These are kind of hybrid supercapacitors. So I'm working with the different divergent fields in which it is covering the various directions. So today I will just discuss on the biosensors and BioFETs by using two-dimensional materials, and I will also discuss a little bit about the possibilities how we can make a kind of biosensing devices or enzymatic or non-enzymatic devices. So most of my work is related with the two-dimensional materials during my PhDs. One of my PhD is on the quantum devices, spintonic and quantum devices, while my second PhD on the optical sensing using two-dimensional materials and some of the high-frequency device applications. So I will briefly discuss about the biosensors, what the biosensor is, and how we can design some kind of sensing devices. The basic structure of the biosensor is that we have analyte, biospecific surface, biosensors, and the output result. If we talk about in a simple way, I guess there are a few students also in this workshop, so I will try to explain in a very simple way that I will not be go into the detail. I will give you the brief overview of the work. because this part actually, the sensor is the sensing device. Before sensing, we need to optimize which kind of analyte we want to detect. Okay, analyte is a part that discuss us and that tells us that we want to detect. Suppose if we are making the glucose sensor, then the glucose is our analyte. In some cases, we are detecting dopamine, so dopamine, then the dopamine will be the analyte. Like if you are using we want to detect cholesterol, then cholesterol will be our analyte. And bio-specific surface is the surface if we are suppose we are using blood, and blood contains glucose, dopamine, as well as the cholesterol. So how specifically we can detect one of the entity of the blood that can clearly, it tells us that the concentration of cholesterol or the dopamine in our blood. So in that case, that we need a biospecific surface that only detect only one entity that we want to detect, and these results can be calculated, can be detected in the form of like computer based outputs. What kind of analytes we can detect in general? We can detect protein, glucose, bacteria, DNAs, some cholesterols, uric acid, dopamine, and some amino acids. These are our analytes that we can detect. What is bioreceptor actually? These are the two or three components that I want to explain. This specific surface, and then we will use the bioreceptor. Bioreceptor will differentiate whether which kind of entity we want to detect from the analyte, or from the any sample. There are different type of bioreceptor we will have, like some of them are enzymatic, some of them non-enzymatic or coenzymatic, some of them will be antigen or antibodies, nucleic acid, tissues, and cells. These are the biorecognition elements, are known as the biorecognition events. Why we need the sensing devices? If we talk about the biosensing, in biosensing we can detect, we can utilize the sensor for food, for medical devices, for agriculture in general. There are other applications as well. We can use for the disease detection, for the environmental monitoring, for soil quality indicator, or for the food quality and water quality, and we can implement in different direction. What challenges we have in the biosensors? The first challenge is that our sensor should have high sensitivity. It should have also high selectivity and quick response. If we talk about the sensitivity, means some of the biosensors are very good for, suppose, for sensing of one of the analyte, but it's not good for the other. So we have to be a very you can say clear, or we should have a very uh, good compatibility of our analyte and our sensing device. So should have a good sensitivity for our target analyte. Selectivity is another very major issue that should be clear. Suppose if we have a sensing device, and it should have the capability to detect, like if we have in blood we have a dopamine, we have um, cholesterol, we have glucose. So how is good our sensor is that can select one of the entity that we want to observe. So that is the selectivity. We should have very clear about the selectivity of our sensor, and response should be very quick. Then we can make a high performance and quick response devices. So for this we need a very good nanomaterials, that could provide us a quick response, high sensitivity, and high selectivity. Second, there should be an appropriate methodology or techniques that could be utilized to make high performance results. I will explain, because I'm just talking about a two-dimensional material. I will discuss with the two-dimensional materials based biosensors and BioFETs. In biosensors, if we want to make any kind of sensor, we have a substrate, we deposit any nanomaterials. Here I am using the graphene as nano thin sheet, and we deposit an electrode on the top of that. Then after that, we disperse the bioreceptor, or known as the enzyme immobilization. After the initial characterization, what we do, we test our device with some of the analyte, and we get the response in terms of current versus time. So this one is a kind of a general procedure of the biosensor, like graphene-based biosensors. What we detect, we see the change in the resistivity, change in the conductivity, or in the current. We can also detect, if we want to make a BioFETs, we can make a kind of we can study the mobility parameter or change in the transition in the electrical transport. This is an overview of the device. Here we can say we have substrate with silicon and silicon oxide, and then we transferred graphene on the top of that electrode substrate, and this hexagonal shape sheet is representing the graphene sheet, and on the top we can deposit any kind of electrode. Mostly we are using the gold electrode. So what the next step is, if you apply the voltage and measure the current in kind of two probe electrode measurement, so we will see the change in current, and when we increase the concentration of our analyte, the value of the current may increase or may decrease depending upon the interaction of the analyte with the surface of the channel. So in some cases, the current can be decreased, in some cases it could be increased. But in the case of BioFET, I will explain how you can transform this device into the FET device. FET means the field effect transistor. This is actually process of how we can make the graphene from the scratch. It means we have a copper file, and methane normally flow through that this chamber, and what will happen, it will decompose in the form of carbon and hydrogen at a specific temperature and a specific pressure, and there is a some amount of argon is also flowing through that vessel. And after that, after like a few minutes, we can make a clear thin film of the graphene, and this process actually is known as the chemical vapor deposition grown graphene, CVD grown graphene. However, we can also make the graphene with the mechanical exfoliation, but for the large scale or the industrial scale utilization, we need large scale devices. For that, we can use the CVD. And then after on the synthesizing the graphene on the copper file, we can transfer, because copper is a conducting. So if you want to make a FET or field effect transistor, what you can do, you can transfer this graphene on any desired substrate by using this method. Here is the graphene is on the copper file. What we can do, we can deposit, we can spin coat a thin layer of the any polymer. Here we are using polymethyl methacrylate, and after that we can etch the copper file by using ammonium persulfate solution, and then we can transfer on the desired substrate. By doing that, we can transfer our graphene film on any kind of desired substrate. This is actually a device fabrication process. Then we can deposit a metal electrode on the top of that, and this metal electrode can work as we can make sensor if we are not applying any bottom gate voltage or back gate voltage. So we can only study the sensing behavior if you are applying the potential at the source and measuring the current at the drain. So this will be a simple sensing device. If you want to make a FET, then what you can do, you can apply the voltage on the bottom side, and you can also see the kind of a transistor behavior. This is optical image of the device that shows that this Hall-bar pattern actually representing the graphene sheet, and these gold and yellow electrode is representing the gold electrode. If you want to study biosensing property, what we do, we can disperse, or we can disperse the analyte on the top of this substrate. Before that, we take the reference, means unaffected device, and after that we can deposit some of the concentration of the electrolyte. We started from the lower concentration, and slowly we can increase the concentration, and we can see the trend, how the electrical resistivity or conductivity changing of the device. So if we want to make the cholesterol, so we need a bioreceptor. Cholesterol biosensor means, suppose cholesterol is our analyte, but our bioreceptor, there should be a bioreceptor that will be only detect the cholesterol components. So this part is known as the cholesterol oxidase. Cholesterol oxidase will be deposited before the our cholesterol immobilization. Suppose this is our substrate. This one is our cholesterol oxidase, is the bioreceptor, and then what we could do, we can detect the desired entity. Suppose we have cholesterol in this case. So this one is a real device image of the optical, same kind of image, that how you can do the measurement. We apply the current, we measure the voltage. We have this purple region is representing our channel, graphene channel, and this one is the gold electrode device measure. This one is our device measurement configuration. What results we get in the case of BioFETs? In the case of BioFET, if we have a graphene, we can also use another 2D materials like MOS2, molybdenum disulfide, or tungsten disulfide, or you can make the combination like a heterostructure. We can also choose this kind of thin layers as channel material. What we will see, on the bottom side we have a gate voltage. On the left side we have a resistivity. We see in the pristine case, what will happen, we have a clear we apply the voltage, means gate voltage on the bottom. If you see here, the gate voltage is now on the bottom. If you see in the more detail, you can see this one is a back gate. We apply on the bottom side, and what we do, we apply the fixed voltage, and we see the fixed current. But when we increase the voltage, back gate voltage, we can say the transition, the electrical the charge carrier concentration will be changing, and from negative, suppose here we have minus 60 volt to plus 60 volt. We see that the charge there is a charge neutrality point, mean carrier concentration is varying from as the gate voltage vary. And at one point we will see that the transition, and that point is known as the drag point, or known as the charge neutrality point. And when we immobilize this transistor, or this channel with some kind of analyte, this is the concentration of the analyte, we can see that the shift in the transition peak, or in the charge neutrality point, in this shift can be clearly observed. And when we increase this concentration, we can see that the change is more prominent. So by doing that, we can also observe the concentration, and we can detect the concentration of the analyte. Means we are sensing that our cholesterol by using the FET device. This change in drag point can be plotted here. How the concentration is affected with the how the drag point is affected with the cholesterol concentration. This is one way that we could study the sensing property. We can also do this sensing directly. Suppose a normal sensing device. What we have, we have a concentration of the cholesterol, and we will see when we apply the potential. Look here, when we apply the potential and we measure the current, and there will be one value of the current for the reference case. When we increase the concentration, and by increasing the concentration, what will have your current should increase or decrease. In this case, where our current is increasing, and when we increase the concentration from 1 mg per ml to 2 mg per ml, our concentration is increasing. So we can make more precise if we decrease the concentration from 1 mg or less than 1 mg. We can make a more precise detection element. So this part is also known as the biosensing, means biosensors. Here we don't need any kind of gate voltage. We can deposit our thin film on any kind of desired substrate, mostly insulating substrate, and then we can just apply the potential and measure the current. And our responses are, you can see the linear response can be observed. So we can also uh, uh, get some information from the previously designed devices, and we can also compare our results with the previously laboratory designed devices. Like already we use a known blood serum sample, and in the blood serum we detect the current for our case. And then what will happen, how we can see that the results that known value was the 40 milligram per ml, and the detection of value was to this value. And when we compared with the previously known value, our result was like around 95% in some cases, and maximum we reached up to 98%, 98.8%. So in this case, we can observe that our device can work for some kind of new devices, and we can also observe that the detection is possible by using graphene as a sensing channel. There is another device. This process is actually known as what we do when we use a bioreceptor before immobilization of our analyte. This process is known as the enzymatic process. We can also do the non-enzymatic process. In non-enzymatic process, what we do, this is a simple method to prepare some nanomaterials. Here we are preparing the cobalt oxide and polyaniline and the cobalt oxide, and we are using cobalt oxide nanoparticles by using hydrothermal method. We're preparing this cobalt oxide nanomaterials, and then we also using the polyaniline as a by making the composite of the cobalt oxide with polyaniline. And cobalt oxide polyaniline nanocomposites can be used for the biosensing. Suppose here we are testing for the dopamine. In this case, this one is actually non-enzymatic process, in which we use electrochemical biosensing. In the case of electrochemical biosensing, I am just skipping some of the basic information of that. That we have electrolyte. In this case, we are choosing deionized water, and what we do, if we do the 3 electrode assembly measurement, in which we have one reference electrode, one working electrode, and one will be it is known as a counter electrode. From the low concentration, we see we do the chronoamperimetry, and in the chronoamperimetry, what will have effect, the same kind of change in current can be observed. This is actually the measurement from the potential state, in which we apply the potential and we measure the current at a different time. Suppose in the 50 second or 75 second, we can see the current is increasing, and after increasing the current, when we increase the concentration in our dopamine concentration in our liquid, that is our electrode is dipped in, and we see the concentration is increased, and the change in current is also increasing. So this case, actually, for this case, we are using glassy carbon electrode as the working electrode, and we deposit glassy carbon, and the cobalt oxide and polynil composite is deposited on the surface of the glassy carbon, on the tip of the glassy carbon electrode. And in this case, we can see that the concentration, we can also see like a linear response, and this is kind of a non-enzymatic method, because we are not using any kind of any bioreceptor. So we can directly detect our desired analyte. In this case, we are detecting dopamine. So the important parameter is the interference study. Because when we want to detect the dopamine, whether how we can decide whether this sensor is useful for the dopamine sensing, because there are, if we talk about the blood, there are some other concentration, means some other analyte like glucose and other analyte is also present. So how we can differentiate from the other analytes? So we could do the kind of interference study that we initially we do with the dopamine, and after dopamine we do the glucose. Means some materials are very specific with some detection of some of the analytes. Suppose this material is more shows very clear performance for the dopamine. In the case of glucose, they are not it is not showing any kind of a change in the current, and we see when we add the dopamine, the result is prominent. When we use the uric acid, we use the dopamine, and we use the glucose, and the result we cannot see any kind of clear change. And if we do in the same thing we have done once again in the same thing, once we did with the dopamine, then we added the glucose, we added the ascorbic acid and uric acid. See, there is no big change, no transition is appeared. I mean, then we did again with the dopamine, and seen there is a clear transition is sensing behavior is observed. Similarly, after that again, means kind of the confirmation that this kind of sensor is more suitable for the dopamine. This process is actually known as the interference study, that this sensor is not well interfered with the other sense analytes. So it means you can make this kind of sensor for the dopamine sensing. So these are the things that we could observe by utilizing and the nanomaterials, and some of the nanoparticles, nanorods can be utilized, thin nanofilm can be utilized, and heterostructure can be utilized by making devices such as BioFETs and biosensors. And some electrochemical biosensors can also be utilized. You can visit my website for the more detail. Some of the recent publications we have published in the various journals is presented here. If you want to look at it, you can visit my website. Thank you very much.
All right, thank you so much for the nice presentation. We have plenty of time for questions. Let's see. We have a question from Syed Shehzadul Hassan. Syed Shehzadul Hassan. You go ahead and ask your question, Shehzad.
Yeah, thank you very much, Dr. Zahir, for such a nice talk. I have two very basic questions. Number one, in last couple of slides, you mentioned very high specific detection of, for example, dopamine in the presence of, there are hundreds of different types of molecules present in the blood.
Yes.
What is really the the chemistry of this specificity. Because in that case, what I understood is you haven't attached any biosensor, it's direct detection. So this was the first question. And the second, even more basic, in the previous part where you use those receptor, for example, for cholesterol to attach.
So bioreceptor, you're talking about bioreceptors.
So what chemistry is used to attach on graphene? It is just adsorbance or some covalent bonding, or
yes, this is kind of a absorption. Like sometimes some binder has been used and to make a well adhesion on the surface, and in some cases we can just deposit on the surface of the, or you can spin coat on the surface of the or in some case we do the drop casting, and depending upon the quality of the polymer that we use as bioreceptor, and sometimes we use some binder to make it well connected with the surface. And second question is the bioreceptor. Yes, bioreceptor, because two kind of studies normally do. One is the enzymatic study, and one is the non-enzymatic study. Yes, definitely there are a lot of different biochemical analytes are available in one of the species. So the idea behind how we can differentiate is like kind of sometime literature helps us to finding the suitable chemical, that means the working electrode for that. And sometimes we have to do kind of a hit and trial method, that which material is more suitable for sensing or for the interference study. Because in some cases we have to make some kind of composites, and to make it useful for specific analyte.
Okay, so we have another question from Dr. Shahid Khan. Shahid,
yeah, all right. Assalamu alaikum, Dr. Zahir. Can you maybe go back a few slides to your cholesterol sensing study? So go back. Yeah, all right, good. And एक और slide जाएंगे, go one more slide back. So first question, yeah, just go back one more. I was wondering about the scale of the device. Yeah, this device. So can you tell us what are the dimensions? Yeah, I see. It says the scale now, I see it's 20 micrometer. Okay. And then you've got the electrodes, and then you've got the graphene in the middle, and then you are coating your receptor on top of it, and then the receptor is binding cholesterol. So in terms of, I'm thinking again of, all right, this could be very useful, right? So now let's think about marketing this. One immediate question that comes to mind is, okay, all right, you've done one assay, you've added your sample, and now can you reset and use your device again? Because if it's just a one-shot device, then we can't market it, right? We want to make a small device that you can use again and again and again, which is also cost effective. So maybe in this, which is a kind of nice pilot study you've done, I think, where you've now shown all right, now if we can take this into the market, the other thing would be the kind of receptor you use. See, the problem with the receptor method is that proteins tend to denature. So that's the other thing we would need to think about in terms of, how do you sort of preserve the protein, maybe sort of embedded in glucose or something. But maybe you can sort of give us an idea. You must have thought about these things, because clearly, again, this is an applied medical physics kind of project. And I presume you want to market these things at some point.
Okay, and I think we must, sir. First one is the about the reuse of the device. If
Bilkul.
we are talking about the biosensors,
Yeah, no, that's what I'm focusing specifically on the cholesterol thing.
yes, actually, what we could do, because in the lab, when we see the devices on the large scale, we can make kind of disposable devices like a chip-based devices, paper-based devices that we transfer graphene on the top of that. We use once and we can throw it out. So the thing is that if we optimize the parameter for that, we could transform in the kind of disposable devices. Number one, because graphene is not a very expensive material that if we see in the CVD graphene, we can easily get kind of paper-based devices and can be utilized. Secondly, yes, your second question was if we talk about the BioFET cases, definitely in the case of BioFET reutilization, sometime it works. We have tried, because if you do the biosensing, it intercalate on the surface of the graphene. So when we clean it with like deionized water or with the methanol, you can just retain your device once again. We could be utilized. So in some cases we could do this kind of you can say utilization could be possible. It means reutilization. In the bioreceptor case, yeah, definitely if we talk about the bioreceptor, in some case, if we use kind of antibodies, it is really expensive. So what we are planning to do right now, we want to design some kind of DNA as a bioreceptor that could be helpful for the that can means we can make the like a low cost devices.
Bilkul. No, that's a smart idea. And I think, okay, your argument, I think of, if you sort of miniaturize it, then the cost comes down. Graphene is not very, expensive, as you say. And yeah, so this is very interesting, Zakir. And I think I have a lot of EM colleagues who have, so graphene is also commonly used in cryo-EM work for grids and so on. And there are all kinds of sophisticated methods now for cleaning it. So let me get back to you on this, but very nice. Thank you.
I will add one more point that recently one of the Korean group has used the graphene BioFET for detection of COVID-19. They have already done with the same kind of
I
procedure
think,
they did.
once you have the principle, but it all has to do with, I think that the detection would, essentially, if you have a specific receptor for X, the X could be COVID-19 or anything. I mean, the principle is the same, right? But it's essentially now to actually manufacture it and market it, it's the cost effectiveness we need to think about. And, how many uses can you get out of it? If it's one shot, how cheap is it? Or can you make it 1,000-shot device and things like that? Great, good.
Thank you very much.
Okay, are there any other questions, especially from the students? Okay, if not, let's. So we have one question from Sana Imtiaz. And the question is, if we are going to use the device, how would we ensure the optimum results? Simply washing with DI water may leave some stain?
For the utilization of devices I have done work with the various biochemicals like urea and L-protein, L-cysteine, and in most of the cases when we clean it, and it works well. But depending upon how good you are in the cleaning, definitely cleaning of the graphene is really a tedious job. But if I have done with the urea, it works well. I did with the L-cysteine, it also works well, because I had a lot of devices and I haven't tried many devices to clean it and reuse it, because I have another device I utilized it at that time when I did this experiment. So it could be possible that we could utilize, because because the molecules of the analyte is not absorbed, not making any kind of covalent bonding. It is actually kind of van der Waal staking at the surface. So if it react with the surface, then it makes problems to detach these biomolecules. But if it kind of surface resorption, or you can say surface entanglement at the surface, so we could reutilize it.
Okay, I thought you have a question.
Yeah, just a quick question. In one of your table, you mentioned some percentage for detection accuracy or something along the line. How did you measure those percentages? I just was
Actually,
wondering.
what I did, I took the data from the laboratory, the people for the concern, means if we have pure cholesterol. And
Yeah.
I took the data from the laboratory. If they do the measurement, and what will be the results if they suppose, I mean, kind of, they said this results is for the pure 40 mg per ml solution, and the results are, these are the results. And then I did the same experiment with our device and compared how deviate from the already utilized device in the laboratory, in the clinical laboratory.
Okay, so you standardize it against a certain maybe
Yes, sir, already market available device.
Okay, okay. Thank you.
Okay. Any other questions from anyone, students, non-students, faculty, staff? Zahir, I have actually a very basic question. So what actually is the advantage of these sensors as compared to what's available in the market? Is it the cost effectiveness? Is it more the use, or kind of, or the, basically at home use that you are looking for? Because cholesterol sensors, I'm sure there are a bunch of them out there that you can use. Why we need another one?
Okay. The thing is, in the initial slides, the main focus is the selectivity, and second one was the high quick response. In most of the cases, in the recently available devices, the response and the detection, I mean selectivity, is the major issue. What we want to do, we want to make a fast response sensing device that could, because graphene is a very quick response, showing very quick response. Any of the analyte that interact with. So because of the large surface to volume ratio of the graphene surface relative to the already available devices, so this can provide a high and quick response, high performance and quick response.
Okay, good to know. Okay, I think we are going to take, so it's our next, which is the last talk